Hello, I'm Zoe Cantor,
Hello, I'm Honey Sunasho.
and we're going to tell you about our Diatom project. We focused on this putative antiporter and mRNA extraction in PCR.
Our project is centered around phytoplankton eukaryotic organisms called diatoms. These diatoms, they do photosynthesis. So one of their main food sources is nitrogen. And then they take in this nitrogen, they undergo photosynthesis, and one of their product is organic carbon. And this organic carbon is then consumed by other marine organisms, which means that these diatoms are one of the very important food sources in marine ecosystems. So because of greenhouse gases and climate change, it is predicted that by the end of the 21st century, atmospheric carbon dioxide is going to increase by a significant amount. So this causes pH levels in the oceans to drop and a rise in temperatures, which can affect the marine ecosystems. So down below, you can see three species of diatoms that we are studying. The first one is Tarsio pseudonana. We actually gave nicknames to these diatoms so they would be easier to communicate. So the first one is called THAPS. The second one is Phaeodactylum tricornutum, or we call it Pheo. The third one is Catastris tennissimus, or we call it CHAT for short.
All right, so as the CO2 levels of the atmosphere increase, so do the CO2 levels in the ocean. So we can see that in this first graph, how, from 1960 to now, the CO2 levels has been steadily increasing. And we can see in the second graph that this fluctuates within each year. So in the summer months, there is more energy from the sun available, and this allows photosynthesizers to do more photosynthesis, where they take CO2 and they convert it into oxygen. And so this then decreases the amount of CO2 in the environment. But then in the winter, the amount of CO2 increases again. So even though there's a zigzag pattern, the trend overall is still steadily rising. And then, why do we call this ocean acidification? So when CO2 enters the ocean, it reacts with water and becomes carbonic acid, as we can see in the diagram at the top. And then this carbonic acid becomes, or kind of dissolves into one hydrogen ion and one bicarbonate ion. And the presence of these hydrogen ions cause the ocean to decrease in pH, meaning it is more acidic. So that's why we call it ocean acidification. All right, I want to quickly go over some of the past research that informs our project. So in 2018, our mentor, Jake Valenzuela and his team showed that T. pseudonana has increased resilience at high carbon. And so these are future ocean conditions around 1,000 parts per million of CO2. So basically, these diatoms under high carbon were able to recover from increasingly drastic exposures to UV radiation. Now, they also analyzed the genes that were being transcripted by these diatoms at low carbon and high carbon. And they found that at high carbon, T. pseudonana, or THAPS, significantly upregulates this putative sodium-potassium hydrogen antiporter. So this antiporter is hypothesized to be really important to maintaining pH homeostasis and potentially linked to T. pseudonana's increased resilience. So the research that we did in our internship focuses on these questions. Are there orthologs of this antiporter gene in other diatom species? And are these orthologs also upregulated in high carbon conditions in these species? And the reason why we're interested in this is because, well, it's great to understand how one species will react to high carbon, but it's really important to look at this issue from a systems perspective and not just the one species, but the entire ecosystem. Because if THAPS becomes more resilient, but other species do not, then that could really change the makeup of the oceanic ecosystem.
As Zoe mentioned earlier, in oceanic conditions with a higher abundance of carbon dioxide, there would be more hydrogen ions released. So what our mentor and his team hypothesized is that DAPS has this antipolar which will pump out hydrogen ions and will take in sodium and potassium ions, which will help regulate the pH level of the diatoms to be in a more optimal level. So if you look at graph A, you can see that in low carbon conditions, which is in dark blue, you can see that the transcript abundance of the gene is significantly lower compared to the high carbon conditions, which is in red or orange above. And if you compare within high carbon conditions, you can see that in light versus dark, the gene is more expressed in dark conditions compared to light conditions. Which makes sense because if you look at graph B, you can see that in high carbon versus low carbon conditions, the gene is significantly more expressed in high carbon conditions rather than low carbon conditions. And if you see in both conditions, you can see that the gene is more expressed in dark rather than in light. So our main focus is that we want to know if other commonly found diatom species have a gene that has a relatively similar function to the antipolar found in DAPS. So what we did was that we ran sequences to find the orthologs of DAPS, and what we found were Pheo and Catastrophes. So this table above shows the orthologs that we found, and you can see that in both Pheo and Catastrophes, there are 2 orthologs each, which means that the orthologs within the same species are paralogs to each other, which means they are related. If you look down here, you can see that there's an asterisk by this gene. This is because we plan to do qPCR to study the gene expression of these orthologs, and which means that we will need a primer. However, we couldn't find a sequence because the gene isn't as annotated as the other genes. So to further support our findings, down here we ran an alignment of all the orthologs within UniProt's alignment tool, and you can see that in the blue sections which are highlighted, you can see that they relatively match to each other.
All right, so here's our experimental design. So we grew these 3 diatom species in both contemporary carbon conditions, so that's low carbon and high carbon conditions. And for each condition, we had a setup kind of like in this picture. So we did 3 replicates, meaning we grew 3 of the exact same species. So here's Nana, 3 Pheo, and 3 Catastrophes. And we grew them in these mini reactors where we could carefully control the amount of CO2 that was being pumped in. And then, yeah, we did this. We grew them for about a week, and then in low carbon, and then we did it again in high carbon. And throughout this whole process, we were saving cell samples. So then we could later extract the mRNA and conduct PCR with primers that we designed in order to test for the transcript levels of the identified orthologs.
So throughout this internship, we all came in with different levels of experience with coding. So we work with the R language within RStudio, and our main objective was to create graphs of the data that we have collected. So we used this program called ggplot2, and well, as the weeks passed, we grew more confident and more fluent in the language. So let's take a look at our graphs. Here we compared the pH of our mini reactors within low carbon and high carbon conditions. So you can see that in low carbon, all the reactors are significantly higher than their high carbon counterparts. However, you can see that there is a big difference in peak within these species. So it is suggested that these peaks are where the diatoms are in their peak exponential phases. So which means that they are taking in carbon dioxide to undergo photosynthesis, and they multiply, which makes sense because when they do photosynthesis, they take in carbon dioxide, which means that when more carbon dioxide is taken from the environment, there is less hydrogen ions within the environment, so the pH is higher.
So every day while growing the diatoms, we took samples and we counted our cells in a hemocytometer in order to calculate an approximate concentration of cells. And then we can make this graph showing the growth curves. So we want to compare how the growth curves of the different species change when they go from low carbon to high carbon. So when looking at VAPs, we can see that the growth curve in low carbon and the growth curve in high carbon looks pretty consistent. You know, they have the same sort of shape, except for in high carbon, it reaches a much greater max value and it just looks like it has greater growth in general. And the growth curves of Catastris in low carbon and high carbon also have the same sort of pattern. Well, they look similar to each other in that they reach a peak somewhere in the middle of the experiment and then they start to collapse. And you can see that in high carbon as well, even though, like in THAPS, it does reach a much greater peak in high carbon. However, when we start to look at FAO, we can see that the growth curves actually change quite a bit from low carbon to high carbon. In low carbon, FAO grew really, really well and didn't really show any signs of slowing down by the time we ended the experiment. Whereas in high carbon, it peaked sort of in the middle of the experiment and then started to collapse. So this shows that a transition from low carbon to high carbon might have a very different effect on FAO than it would on VAPS and Catastris due to its genetic makeup. We can also look at the carrying capacity of the different species. This is the max concentration of cells that it reached. And you can see for Catastris and THAPS, the red and the blue, that the carrying capacity significantly increased from low carbon to high carbon. However, it's more tricky with FAO as the carrying capacity is within sort of the error bar, so we wouldn't really know. You know, there isn't a statistically significant result as to if it increased or not. And from this, we could hypothesize that, well, other species might become more prevalent, FAO could stay the same, and leading to it being less prevalent in the end in a more acidified ocean. All right, so once we were finished with growing and counting our cells, it was time for RNA extraction and PCR. This process had so many steps to it, and we, along with the help of Christine, this undergraduate intern that we work really closely with, we designed protocols to carry out all these various steps. And then when we did them in the lab, we took notes based on our mentor's feedback, and then added the notes back into the protocols to keep improving them. So the first step was to take the samples that we put aside as we were growing the cells and extract the RNA. And for this step, we chose to extract it from the samples grown in low carbon because we wanted to test it out first at that stage. So we extracted all the RNA using the Trizol method. Next, we had to purify the RNA, and we used this RNase spin column, and we also added the enzyme DNase to digest any contaminating genomic DNA because we only wanted the mRNA, because we wanted to measure the transcript levels of these genes. The next step was to do reverse transcription from the mRNA to cDNA. And this is important because PCR, which is the next step, needs it to be DNA, not RNA. So then we performed PCR using primers that we had designed using NCBI Primer Blast. And finally, in order to see our results, we ran gel electrophoresis. Speaking of our results, so the point of this slide is to say that we basically failed the first time. But before I can get to that, let me just give you a crash course in what all of this means, because I know this is a lot on this slide. So what we did is we put different samples into each of these wells at the top. And then if you're not familiar with gel electrophoresis, what happens is a charge runs through. And since DNA is negatively charged, it runs through. But since we have this in an agarose gel, the smaller fragments of DNA can get through really easily and they run much further through the gel, whereas the longer segments get stuck sort of near the top. And from this, once it's done running, we can look at it and we can use the location of the bands to see what the length of the DNA in those samples were, and what the length of the DNA that we amplified using PCR. And this can tell us if our PCR successfully amplified the right genes or not by comparing it with the expected PCR product length, which is in this table. All right, and we ran this in triplicate, meaning we basically did the same thing 3 times. You can see 1 is replicate 1 in pink, replicate 2 in green, and 3 in orange. So now on to why we failed. First, we added positive controls where we put primers in for genes that we knew should be in our sample, actin and rubisco, very common and important genes that would be transcribed. Those were in those wells. As you can see, not a band in sight. What that basically tells us is we did not have enough DNA from FAPS, because that is the species that we used for those samples. So already off to a bad start, but even more confusingly, if we look at these 3 bands, these are in our well for Cathoceras with the primer for the ortholog we identified in replicate 1, 2, and 3. Keep in mind, this should be the exact same. We put in the exact same DNA, we put in the exact same primers. However, you can see those 3 bands are in 3 different spots, meaning a different gene must have gotten amplified in each of the 3 replicates. We have no idea how this happened. Our mentors don't know. We asked like a graduate student in the lab. He doesn't know. Anyways, so after we did this, we decided we needed to rerun our PCR and gel electrophoresis to see if we could get better results. This is our second attempt. We changed it in a couple of ways in order to simplify it. So we only did 2 replicates. And we increased the amount of cDNA that we added to the samples before going to PCR, because we thought that maybe a problem with the first one was that there wasn't enough cDNA. And we also increased the volume of primers, but we only did that in replicate 2, because we weren't sure if that would help or not. And the final thing is we did not amplify Theo this time, because we wanted to keep it really simple. Okay, so what did we find from this? First, all of this stuff, that's primer dimer. Basically what happened is our primers, instead of binding to the gene, some of them just bound to each other, causing a bunch of these really short fragments that flowed all the way to the bottom of the gel. So those can basically be ignored. Then you might be wondering what's going on over here with Cathoceras. Oh, and I should mention that I'm mostly analyzing the second replicate just because since there were more primers, it makes it a bit easier to see what's going on. But all of this does apply to the first replicate as well. So in this Cathoceras, there's a case of non-specific binding. Basically, the primer that we designed, instead of just binding to the one gene that we wanted to amplify, it bound to a bunch of areas in the mRNA, which caused a bunch of different genes of different lengths to be amplified, meaning that there's not one distinct bar at a specific length. So that basically means that we would need to go redesign a primer to make it more specific and try again. So Cathoceras' results are also not super valid. So these 2 are positive controls. This time our positive controls did work. We have the bands in the right places. You can compare C in the table, it should be at 162 and 332. And if you look at the key over here, it is indeed around 162 and 333. So that was great. It does mean that we had DNA this time. So our last well is this STAPS one. And you might be thinking there's only primer dimer. There's none of the actual expected gene length, which as you can see in our table would be around 287. And that's right. There is none in that area. So why isn't there any of the antiporter? Well, the DNA, the samples that we used to extract our RNA from were in low carbon samples, and they were taken in the daytime. And if you think back to this chart that Honey showed you like a while ago, you can see that the expected transcript levels of this gene are really low at that period, which is in the daytime at low carbon. So that's probably why we didn't see any. And you might be wondering, why did you pick the low carbon ones to sample if you weren't expecting to see any? And that's because we were really just trying to test out our method, but since we ended up having to redo multiple steps, it took a bit longer and we didn't actually have time to do it at high carbon. Okay, so unfortunately, our internship is coming to end and we didn't quite have time to go and do the process for the samples at high carbon to see the results. However, our mentors, our Christine, the undergraduate who we work with, they will still be doing that. And I'm really excited to see the results that they get. So the future directions for this project are to improve our various protocols for mRNA extraction, purification, and reverse transcription. Also, like I mentioned, to do mRNA extraction and PCR on the high carbon samples. And then ideally, we would perform reverse transcription quantitative PCR, RT-qPCR. And this would allow us to measure the transcription levels of the antiporter orthologs in a really accurate way, just through adding this special dye to the PCR rather than having to run a gel. It's a really interesting process that I won't get into right now. After this experiment, we would still have more directions to go in because we'd love to find if these findings could be replicated out in the field through a potential mesocosm experiment. But that would involve developing a process to measure antiporter expression levels from a field sample, which would be much harder because instead of just having one species of diatom in a carefully controlled reactor, it would be the ocean with all sorts of different species of organisms. And then just in general, it would be amazing to have more research into this antiporter, figuring out if the orthologs in the other species are also localized to the plastid membrane using tagging, or using CRISPR to knock out the functionality of this antiporter and then see how the diatom reacts, which would give us many more clues into the function of this antiporter.
Apart from conducting experiments within the lab, we are also collaborating with the SEE, or System Education Experiences team, and high school teachers to develop a curriculum for high school students. So what we're trying to do is we are trying to demonstrate the concept of resilience, which is how much a system can adapt to stressors, and we would measure their recovery rates, which would then relate to the concept of tipping points, which is how much a system can withstand a stressor before they fall or collapse. So we attended weekly meetings, and within these meetings, we brainstormed activities and analogies which would help, well, us and other high schoolers understand this concept better. So one experiment that we all agreed on as a team was something called the penny experiment. So students will be pipetting out water droplets on a penny to test the water surface by measuring how many droplets the bubble can withstand before it spills out. So other activities also include a catapult and punching stand. So depending on how much or type of stressor that is put on, we can measure their different recovery rates, which can then show that in these systems, there are many factors that affect tipping points. However, these systems aren't only just ecological systems, they can also be systems around us, like political systems or transportation systems.
All right, and before we end this presentation, we just want to say a couple of words about the experience of working at ISB as high school interns, because it's been really amazing coming in here as a high schooler, getting to work in a scientific institution. And if I had to name one takeaway, I think for me it would be about failure, because when you're a high school student, you're doing research projects for your school, you're reading scientific papers, and you're just seeing success after success after success. You're not seeing all of the trial and error, the mistakes that went into the creation of those papers, and you're not seeing all of the unpublished papers, that the projects that never even succeeded at all. And And so coming into this institution and then just making mistakes, like, you know, breaking flasks or pipetting the wrong amount or spilling diatoms, um, at first it was like, oh my God, like I'm like messing up so much, like this isn't what happens. But what I realized is this is what happens. Science is all about like messing up and doing it wrong and having to redo your thing and maybe hypothetically, like running a whole PCR in gel only to realize that you must have like pipetted the wrong primers 3 different times. Like, like, and that's okay. That's a part of the scientific process. And if you don't take risks and make mistakes, you're not going to have findings. And that was definitely something that I learned that I think is really valuable to me.
Well, for me, well, me and Zoe have never really been in a professional lab setting before, and we are very grateful that we have gotten this experience. However, well, science is hard. There are many concepts that like intertwine together and well, these systems, and it can be hard to like untangle and absorb everything. So one of my main takeaways is about learning. So STEM is a field that requires collaboration, and I would say that things tend to go smoother if we are working in teams, which is well a team of 3. Yay!
That's Christine, by the way, if you're wondering. We keep
Yeah.
talking about her.
So yeah, so my main takeaway is learning. So if you don't understand anything, you should just ask. You shouldn't keep it to yourself because that's not gonna help anyone or help you at all.
Yeah.
So yes, so I would say that you should advocate for your own learning. Just don't be afraid to ask questions because that is how you learn. And there also might be someone in the room that has the same question as you and may just be as confused as you as well. So be more open-minded and be happy to learn. Last but not least, we would like to say thank you for everyone who has supported us throughout these 7 weeks of our internship. The Belika Lab and Diatom team members, Dr. Belika, our mentors, Jake and Chris, and our fellow lab interns, Rihan Ashwin. We would also like to thank ULC team, which made this internship possible in the first place, or else we wouldn't even be here. Claudia, Miranda, Becky, thank you so much. And we would also like to thank you, our lovely high school interns who have always been with us and support us throughout these 7 weeks.
Yeah, thank you so much to everyone and have a good day. Bye.
Bye.